Hello, and welcome to our presentation on Pacific Biosciences single molecule real-time sequencing, or PacBio-SMRT for short. PacBio-SMRT technology was created by Pacific Biosciences and was based off research performed at Cornell University. PacBio next-generation sequencing has been around since 2010, with SMRT technology being commercially available in 2011.
The template used in PacBio-SMRT sequencing is called SMRTbell. This template is a closed single-stranded circular DNA that is loaded to a chip called a SMRT cell. The SMRTbell diffuses into a sequencing unit called Zero-Mode Waveguide, or ZMW, which provides a small available volume for light detection. The ZMW is a nanophotonic confinement structure that consists of a circular hole in an aluminum cladding film deposited on a clear silica substrate. Each SMRT cell contains 150,000 zero-mode waveguides.
A different fluorescent color corresponds to each base on the terminal phosphate. Each time a new base is incorporated by the polymerase, a pulse of light is created by the tag. The ZMW detects this pulse and calls a base for the unique wavelength created by that pulse.
The attenuated light from the excitation beam penetrates the lower 20 to 30 nanometer of each zero-mode waveguide, which gives the most powerful light microscope with a detection of 20 septoliters, or 10 to the negative 21st.
Again, as each base is added, a unique wavelength is emitted by the fluorescent tag that is picked up by the ZMW. This process continues until the synthesis is complete.
The graph shows the limitations of PacBio-SMRT sequencing. The longest read PacBio is able to read 60 kilobases. The most common read lengths, however, are 10 kilobases.
Some of the pros to PacBio are long reads, tens of kilobases, high accuracy to 99.999% of Sanger quality reads, uniform meaning we have no GC bias, single molecular resolution, which requires no PCR amplification, epigenetics, both RNA and DNA read lengths with 99% single molecule accuracy. Some of the cons are not high throughput. Some of the wells do not possess or have DNA templates. It's rather expensive with a high error rate of about 12%.
So some of the differences between Illumina's technology and PacBio's are the average read length are different between the two. Illumina generates reads of about 150 to 200 base pairs, while PacBio's reads can go up to 30,000 base pairs. Illumina is less expensive at $502 per megabase versus PacBio's $2,000. PacBio uses single DNA molecules to do the sequencing, where Illumina creates clusters of DNA. Fluorescent markers are attached to the terminal phosphate in PacBio. Illumina attaches the markers to the sugar base. Polymerases are anchored to the bottom of the SMRT cell in PacBio. Illumina has free-floating polymerases. PacBio is much faster at 2 hours per run versus Illumina's 27 hours per run, and Illumina has a much lower error rate from the much shorter reads.
Some achievements of SMRT sequencing include producing highly continuous assemblies of eukaryotic and bacterial genomes, discovering short tandem repeats and mutations associated with genes and other forms of diseases, discovered gene deletion breakpoints in 6 cancer cell lines, and characterized structural variants in the human genome.
Some of the achievements in research for transcriptome research include identifying human intron structures previously unknown and unannotated, characterization of alternative splicing sites used in cellular blood component production, as well as identifying novel isoforms in the HESC transcriptomes using hybrid sequencing.
Some achievements that PacBio has done from epigenetics research is discovering new M6A and M4C MTAses and methylation patterns in 6 bacteria, detected M6A and M5C residues in E. coli-deduced target sites of MTAses that catalyze M6A modification, identified virions favored genotype-dependent motifs in Heliobacter pylori, detected intracellular heterogeneity in genome DNA modifications in Streptococcus pneumoniae.
Finally, in a paper published in 2019 by Eichler, PacBio's long-read sequencing technology was used to create reference genomes for genetic diseases that previously did not have one.